Hello, my name is Adrian Green and I'm currently a postdoc in the lab of David Reif at North Carolina State University. It is my pleasure to talk to you today as part of the 2021 ISMB ECCB scientific meeting about leveraging high-throughput screening data, deep neural networks, and conditional adversarial networks to advance predictive toxicology. There are currently 85,000 chemicals registered with the U.S. Environmental Protection Agency that are manufactured in or imported into the U.S. Of these, approximately 4,500 have rigorous toxicology data, leaving over 80,000 chemicals untested. To address this huge data gap, high-throughput screening and computational methods are vital. Computational approaches to bridge this data gap have been developed utilizing chemical structure, with quantitative structure-activity relationships and read-across being the most common. Recent advances in machine learning, such as deep neural networks and conditional adversarial networks, or CGANs, offer an excellent opportunity and have not been thoroughly explored. As part of an ongoing effort by Robin Tangwe at Oregon State University to screen chemicals for neurodevelopmental effects, 32 zebrafish embryos were exposed to over 1,000 chemicals at 6 concentrations from 6 hours post-fertilization to 5 days post-fertilization, as shown in the diagram on the left. These zebrafish were then assessed for mortality and 17 physical defects and abnormalities at 5 days post-fertilization, some of which are shown in the image in the bottom right. This resulted in a massive data set containing information on over 200,000 individual zebrafish and a wealth of toxicity data. While the 1,059 chemicals in this data set are only a small fraction of those found in the Integrated Chemical Environment, or ICE database, they represent a broad range of physical-chemical properties as shown in the principal component plot on the left. Further, the plot shows the physical properties of the chemicals found in our training and test data sets, with the test set coming from a recent follow-up study conducted years after the initial high-throughput screen. Now, to fully leverage the chemical and toxicity data in our database to make toxicity predictions for untested chemicals, our analysis utilized a novel algorithm developed to map and vectorize 3D chemical structure that was originally created for use in material sciences. This method, shown in the box on the right, converted the 3D structural information from PDB files into a weighted set of points that retain all the information about the original chemical structure. This structural information was used as input to train 2 types of neural network generators. The first utilized 2 deep neural networks in series to determine the relevant chemical information and generate a toxicity matrix for the 18 mortality and morphological endpoints. This generated matrix was then compared with the empirical toxicity matrix as shown in this diagram, with the darker matrix shading indicating higher toxicity values. The network was trained to minimize the difference between these 2 matrices. In a similar manner, we utilized a conditional generative adversarial network architecture, which takes the chemical structure information as input. The weighted set of views were then passed through 2 fully connected neural networks to produce a predicted toxicity matrix. The chemical features, along with the predicted or empirical matrices, were then passed to a discriminator, comprised of a third fully connected neural network, trained to distinguish between real versus fake matrices. Figures A and B in the top left show the training loss plots, with the GAN in panel B requiring significantly more training time compared to the deep neural network in panel A, titled GoZT. Following the training, both models were compared against other common machine learning methods trained using the same toxicity data, but utilizing 2D chemical fingerprints as input. The results in Table 1 show that GoZT outperformed all other types of network architectures measured using Kappa and Auroch, while the GAN-ZT underperformed all the other methods. We next investigated whether an ensemble approach combining 2 of the models in Table 1 resulted in an improved performance. Model combinations between GoZT and GAN-ZT, SVM, MLP, or random forest were assessed for improvement, with particular focus on positive predicted value, Kappa, and Auroch. By combining the predicted results of GoZT and GAN-ZT, we were able to improve these 3 metrics to 71.4%, 0.637, and 0.837 respectively. As shown in the Venn diagram, the resulting model removed 4 positive values from the GO-ZT results and 20 false positives were eliminated from the GAN-ZT results. Our results show that deep neural network-based generators are capable of predicting zebrafish toxicity utilizing a novel 3D chemical structure algorithm. Our deep neural network generator greatly outperformed other machine learning methods as well as our own GAN generator. But a combined GAN and deep neural network ensemble model resulted in large improvements in positive predicted value, kappa, and OROC, resulting in a good predictor of empirical zebrafish toxicity. Now that we have our trained model, the next steps include creating a priority list of chemicals for in vivo testing and empirically validating these results. Another avenue that we're excited to investigate is the development of a deep learning methodology to evaluate mixtures. Finally, I would like to acknowledge my postdoc mentor David Reif for supporting this project, as well as our fantastic collaborators Robin Tanguay and Lisa Trong at OSU. I would also like to thank the members of the Reif Lab for all their insights, and the NIEHS and the NCI for funding this research. Thank you for your attention.